Now, Jacob Koruth doesn't need an introduction either.
All right, so my task is to go over some preclinical data with the field medical catheter. And I think as we delve into what the next catheter that's going to solve all our VT problems, you really need to look at what's out there. And the way I think about catheters for VT is you have large focal tips and small focal tips with PFA. And then we have large focal tips with RFA and small focal tips with RF. Before I talk about this slide, there's a few concepts that I think you need to appreciate. We've, for a long time with radiofrequency, assumed, because we lived in the small focal tip world, that contact force was the solution for depth. And that's because our generators were stuck at 50 watts or maybe 70 watts in Europe. And what small focal tips do with high contact force is to compress tissue. We have a publication that should come out in a few months that shows that with small focal tips, you can compress the tissue about 2 to 3 millimeters, and that buys you extra depth. The same rule applies with PFA with a small focal tip. So if you have a 5 millimeter lesion with PF and you compress the tissue with a small focal catheter, you can get to 7. With large focal tips, like the lattice tip, you really can't compress tissue. When you increase force, the cage expands a little bit, compresses a little bit. It engages greater width, but it does not compress tissue. So you're largely restricted by the generator, the output, and the field. So if the catheter can do 5 millimeters with light contact, with a lot of contact, it can go to 5.5. And these fundamental differences is how the current field is stuck at. And if you look at all the publications with focal tip PFA and large tip with PFA, they all range between 5 and 8 millimeters, and that's with repeated applications up to 4 applications a site. And I think what field is going to do is to transform how you make deep lesions. It's going to say that even though it's a small focal tip catheter, its field is so big that it behaves like a large focal tip catheter. So you don't need to be constrained by moving a large focal tip catheter, which even though it's inherently more stable, it's difficult to get into nooks and crannies like behind the papillary muscle. So field medical with their large lesions allows the benefits of a large focal catheter to be brought into the small focal tip catheter. At the same time, you can still compress tissue, which I think is only relevant in healthy tissue. In scar tissue, there's only so much compression you can get clinically. So there is a disconnect between preclinical studies that say contact force causes a lot of increase in depth, both for RF and PF. This is not going to be realized in scar tissue, which is why the answer to making big lesions has to be not the catheter design in terms of small and big, but a generator that can deliver an electric field that's really big. That said, I'm going to show you maybe 2 or 3 slides to give you a sense of what this catheter can do. What you see here is a footprint of a small focal catheter that looks exactly the same as the large focal catheter that we've used in the ventricle. These lesions are 15 millimeters wide, 22 millimeters long, and in both these sections, one picture is with 1 application and the other picture is with 5 applications. So I heard some discussion about, I really want to control lesion size, and there are different ways to do it. You either cut the output of the generator and deliver a shallow lesion, or you maximize the output and get a deep lesion, or you use a single application and get to 10 millimeters depth, and you give 5 repeat applications and get to 15. So there are different ways how you can modulate depth. So, and I think this is important because when you're at the border of the scar, an area that we think is, at least in ischemic VTs, to be responsible for some arrhythmias, you don't want a lesion that spills over into the myocardium by 5 millimeters and take out unnecessary healthy myocardium. So some amount of dose titration is going to be the future of all focal catheters. And there is one way to do this with this particular technology. On the left is an example of a large epicardial lesion. Now, this is not a lesion that was placed in the epicardium. This is actually the epicardial footprint of an endocardial lesion. And what Vivek showed in the previous talk is when you give endocardial lesions, you get this footprint of low voltage in the epicardium. And while there's this big discussion about maybe all you did was to take out a channel, and that's why you lose the epicardial signals, when you see this picture, you know that the reason why you're losing epicardial potentials is not because you took out a mid-myocardial channel. It's really because the electric field is penetrating 10 to 15 millimeters deep that's causing this lesion. In the middle panel, you see a pretty typical PFA lesion histology. And on the right, you see an example of an everted left ventricle. So it's a little bit stretched out. But when you look at that footprint, that is not the footprint that you see with the regular 3.5 millimeter tip catheter. All the preclinical data with all the catheters that are in that range show you one thing. The lesions are about 8 millimeters wide, can get about 5 millimeters deep, and as you increase force and you repeat applications, you do not impact the width, you impact the depth, and you add about 3 millimeters to that. So all the catheters that I know out there can go from 5 to 8 with contact force of 40 grams. But I don't think the answer to fixing VT is to tell everyone that each lesion of their application needs to get to 40 grams. It's not going to be a reproducible solution. The answer is going to be a bigger, easy to deliver, quick lesion. And I think field in some ways is going to get us to that position. This is data from our center. And what we're basically showing is even with this tiny 3.5 millimeter tip catheter, this is not a 9 or 10 millimeter wide catheter, you're getting diameters anywhere from 20 millimeters to 25 millimeters, whether you do one application or 5 applications. With a single application, you average about 10 to 11 millimeters in depth. With 5 applications in this data set, you're seeing 12.5, but some of these lesions were transmural, so we don't really capture its entire potential. So I think with this technology, we, for the very first time, have an opportunity to make really large lesions. And while there is this concept that large footprint catheters can make big RF lesions, I still think, based on the biophysics of PFA, there is no better technology than pulse field to ablate scar. We know that the electric fields are able to go through the scar. It's especially good when surviving muscle bundles are caught within scar fibers. So the answer to me is not pursuing radiofrequency that makes really big lesions. The answer is going to be making rapid lesions with pulse fields because the safety profile is going to be better. If we were to do a pulse field bipolar application for a big, thick substrate, you can make a continuous lesion. We've shown that. But you will never have to worry about char. You will never have to worry about steam pops. You will never have to worry about ventricular septal defects 4 weeks after you deliver radiofrequency. So in my mind, if there was one technology that's meant to solve VT ablation, it's pulse field. And the answer is to have a powerful catheter and generator that does more than what current catheters do. That's all I have to say. Thank you.